This generated sequence illustrates a core objective of bioinformatics. When the system isolates localized matching clusters of genetic data between organisms, it identifies homologous sequences. Identifying these homologies is the explicit goal of sequence alignment, allowing researchers to infer shared evolutionary ancestry and biological function. Mathematically, the ideal way to secure these matches is through dynamic programming algorithms. These equations calculate every possible structural permutation to guarantee a mathematically perfect alignment between two sequences. But modern biological databases now store hundreds of billions of base pairs. Running exhaustive dynamic programming across databases of this magnitude creates a hard computational bottleneck. The time and processing power required to calculate every possible match pixel by pixel are entirely intractable. Researchers require actionable data to trace outbreaks, develop therapies, and map genomes in real time. The physical limitations of computing perfect math actively impede this biological discovery. To achieve genomic analysis at scale, the reliance on mathematical perfection must be abandoned in favor of a strategic, high-speed compromise. The industry-standard solution to this computational bottleneck is the basic local alignment search tool, known universally as BLAST. BLAST is a heuristic algorithm. Rather than calculating guaranteed optimal perfection, it deploys computational shortcuts optimized specifically for speed and efficiency over absolute exactness. This side-by-side diagram illustrates the mechanism. While the traditional exhaustive grid search on the left slowly grinds through every variable, the BLAST algorithm on the right drastically reduces compute time by scanning only for short, localized regions of high similarity, called seeds. Once it locks onto a viable seed, it extends the alignment outward. The BLAST suite comprises 5 distinct programs, each engineered to process specific biological data formats. Memorizing these 5 tools in isolation is inefficient. To accurately leverage BLAST speed, researchers rely on a strictly logical matrix determined entirely by 2 variables, the user's query sequence and the target database. Genomic search relies heavily on 2 primary data formats: nucleotides, which constitute raw, unannotated DNA sequences, and proteins, comprised of functional amino acids. We map the tools on a simple 2x2 grid. The y-axis represents query sequence, nucleotide or protein. The x-axis is target database, nucleotide or protein. The first quadrant maps a nucleotide query against a nucleotide database. The tool for this DNA-to-DNA alignment is BLASTn. BLASTn is deployed for rapid genome mapping, initial gene discovery, and establishing direct evolutionary lineage by comparing raw DNA sequences between species. The second quadrant handles the other direct match, comparing a known protein query against a protein database. This domain requires BLASTp. BLASTp is essential for structural biology. Researchers infer the functional role of an uncharacterized protein by identifying homologous proteins with known structural motifs. Direct domain comparisons are computationally straightforward, but they are entirely insufficient for bridging the biological gap between a raw strand of unannotated DNA and known biological machinery. The remaining empty quadrants represent cross-domain comparisons. A biological roadblock prevents direct search here. Raw nucleotide sequences cannot be directly queried against protein databases because the underlying data languages do not match. 3 nucleotides encode a single amino acid, meaning the raw code must be processed before it can be read functionally. Bridging this gap requires an immediate computational step, known as in silico translation. This diagram illustrates the 6-frame translation mechanism. A single strand of unannotated nucleotide data is passed through an algorithmic filter and computationally split into distinct parallel streams of translated amino acid sequences. Because DNA is double-stranded and a sequence can begin reading at any of the first 3 positions, the algorithm must test all 6 possible reading frames, 3 forward and 3 reverse. We apply this translation logic to the third quadrant, taking an unannotated nucleotide query and targeting a functional protein database. BLASTx is the specific program that translates the query sequence into all 6 reading frames before executing the search. Researchers use BLASTx to discover novel protein-coding genes buried within newly sequenced, unannotated DNA. It is highly effective at detecting frameshift mutations. By analyzing exactly where the translated reading frames fail to align with known functional proteins, researchers can isolate the exact location of a genetic error. A displaced nucleotide alters the reading frame, and the complex molecular structure abruptly twists and misfolds into a disorganized, non-functional state. BLASTx transforms rote genetic code into searchable functional data, allowing scientists to predict biological utility from raw sequences. The final empty quadrant holds the reverse scenario, possessing a known protein query but needing to search a vast, unannotated nucleotide database. This requires BLASTn. The computation here is inverted. Because the database is raw DNA, BLASTn must translate the entire massive nucleotide database into all 6 reading frames to match the single protein query. This computational load is required to find distant homologs across poorly annotated species, where researchers have sequenced the genome but have not yet mapped the proteome. This diagram tracks the extreme edge case. We are comparing a nucleotide query to a nucleotide database, but we are translating both sides of the search. 2 separate raw data streams enter the translation block, and both split into 6 frames before comparing. The tool that executes this dual translation is BLASTx. Translating both the query and the database into 6 frames creates massive structural complexity, making BLASTx the most computationally expensive program in the suite. But this dual translation serves a precise evolutionary necessity. It identifies conserved coding regions between highly divergent species. Millions of years of mutation have rendered the raw DNA unrecognizable at the nucleotide level, but the essential amino acid functions have survived. It is explicitly utilized in non-coding RNA analysis to hunt for completely hidden, unannotated protein-coding genes. By utilizing rigorous computational translation, BLASTn and BLASTx expose ancient evolutionary relationships that remain completely invisible to a direct nucleotide-to-nucleotide search. Generating an alignment through a heuristic shortcut requires an equally rigorous method for validating that the result is real. In a database containing billions of base pairs, random chance guarantees that false positive alignments will occur. A mathematical match does not guarantee a biological relationship. To filter the noise, BLAST scores alignment validity using a probabilistic metric known as the expect value, or E-value. This data plot demonstrates the inverse mathematical relationship of the score. A lower E-value strictly dictates a lower probability that the observed sequence match occurred purely by chance. As the threshold lowers, background noise is filtered out, leaving only high-confidence data. An E-value approaching 0 indicates an alignment so statistically improbable that it separates genuine evolutionary homology from mathematical coincidence. A fast heuristic alignment is mathematically useless without this rigorous probabilistic validation to verify its biological reality. The reliance on speed introduces specific limitations. Because BLAST is heuristic, it will occasionally bypass the absolute optimal alignment in favor of a fast, highly accurate approximation. The algorithm also possesses input limitations, yielding notoriously unreliable statistics and poor performance when processing very short query sequences. Additionally, E-values generated from low-complexity regions, sequences with highly repetitive genetic code, can easily mislead researchers, requiring manual human review to confirm the biological context. The BLAST suite functions as a modular 2x2 translation engine. Operating it successfully requires matching the specific biological question to the correct query database access. Navigating the BLAST matrix allows researchers to sift through billions of base pairs, pinpointing the functional segments that define an organism's biology.